Full time, so yeah. How do you want us to turn in the Toledo's new paragraph?
I joke around so much, like it has,
Like what I'm thinking. Let me know if it's working.
Oh, well, I didn't see it earlier, but I can check it.
Okay. Thank you.
she came in and said, hey, how you doing? And I was still like, cause I bounce around so much. Yeah, and it looks like I'm barely done, but I'm like, I'm like 90% done. I just was like bouncing around.
Oh my god, did we get stairs? I'm glad. I ate it after school.
I'm glad we were like standing there, and Dr. Sain's just staring at us. I'm like, wait, yeah. Like we're trying to get.
I am deaf if I'm looking down and like working. I thought you had headphones on. I wasn't using them.
I felt like because I don't want people talking loudly. No.
I'm deaf if I'm like looking down or writing something down or like
I know. I didn't have any safety precaution on it. I'm like, I don't know if I can bring food in the lab. So I was like, I can't go in. I was just gonna throw my shoe at you and hope I didn't knock anything in the process. Because, what's that? What's your...
We were doing like crazy labs. They're just reading things.
What lab is this for again?
It was like
What class is this for?
Oh, Kim too. My credits just didn't transfer properly, so I've already taken Kim, and I've got to redo it.
What do you have for the lecture?
Practical.
I love practical. I love it in my class with her. I know you can scout.
Don't even start.
I've never heard scout.
If you, I will actually guess, but take it.
And like you mash up like.
It's such a fun class. I love it.
Just
I feel like they offer next every other spring.
So they'll offer. And like find out. Oh, I want people to take it then. I other spring, so not next spring, graduating.
I'm excited to take it. Oh my god. She's like, okay, I'll sign you in. I was so happy. It was like Christmas.
I had missed Thursday.
I only have 1 year left. I have feeling.
And I'm like,
That's what I was asking you today.
I was like, okay, tell my teacher I'm not coming.
No.
My 3 o'clock now.
I have to try something. So, they have to like pop up, and there's this one, it makes like homemade values to help pay for it.
Well, I told her, I was like, tomorrow.
And that's the one where I showed a,
Like, if I have a quiz,
I can't name one.
I just found the right thing, and
And she did, she had this one, she was just different schools.
gonna give me so fun. You're flying at 80 miles an hour.
Yeah, we're 80.
Yeah, they go at 80 miles an hour. Because you, there's like running. Let's see what she makes of stuff.
Because how many miles we're in today? 4.23.
US1
It was 4. What position do you play?
Really, after this, they still run a lot. Like they're every day, so we do it every day, so it's just like fun.
Well, good afternoon, everyone. How's everyone doing?
Great. How are you?
Good, good. Now, yeah, I know you're in all the other courses. At the beginning of all your other courses, there's an instructor say that, ask you the same thing. It's like, how's everyone doing? That's what they all say, pretty much. I know, I need to kind of work on that. Work on something different, right? Okay, so today's Wednesday, February 11th, and yesterday was when we had our guest speaker from the Toledo Zoo come in, and going to that presentation was like a substitution for our lab assignment. So I know a number of you weren't able to attend, just because you had other things already planned for Tuesday afternoon. I did record the session. So the recording is on the Blackboard site under the lecture recordings. So if you go to Blackboard under the lecture recordings, you're going to find the recording for February 10th. It's labeled as pied tamarin, which is the name of the primate that was the focus of the conservation work. So if you weren't there yesterday, please watch that recording and then, write down like one paragraph about what you learned or what the presentation was about. And then hopefully I have it set up so that you can upload it through Blackboard. I don't know, has anyone tried uploading anything there yet? Yeah. Okay. So let me know if that upload is not working. Yeah.
Does that also apply for the people who also attended, or is this for people who did not attend yesterday?
Right, if you attended, then you were, that's all you needed to do was to be present and you know, like you asked questions, for example, that was fine.
Okay.
So, so for those who did attend, you don't have to write a summary, upload anything. I should have your points already added to the Blackboard grade book. So, you know.
Oh, so, am I hearing that there's already an extra credit for that, or?
No, that presentation actually counted as the lab assignment.
Okay,
So that was not for extra credit.
okay.
But if you went to the card making event on Monday, that was for extra credit. I think I still have the original extra credit opportunity. Someone help remind me, what was that first one?
Pre-med, the Oregon.
Oh, yeah, the Gift of Life Michigan, yes, thank you. So if you haven't done that one, and that is still an opportunity for you if you want that extra credit. Doing the Gift of Life Michigan extra credit. Okay, so maybe keep that one in your back pocket when we get closer to the end of the semester and you need a few more points there, right? All right, good questions, good questions. Any other questions? All right, you know, we came very close to the end of chapter number 5, so I decided, well, let's just move on to chapter 10. So we are skipping a number of chapters there, and we're going to next continue our learning by learning more about how these microorganisms are identified and classified. So that's the topic for chapter number 10. All right, so I imagine you learned about taxonomy before, like usually when you first take, you know, introductory biology classes, you kind of get a survey of different kind of living organisms, and then they usually tell you about how they're classified. So, you know, a lot of this will be a nice review. The understanding here is that all living things on the planet are basically treated the same way. So just because these organisms are small doesn't mean that they're not identified, they're not classified, they're not named. They are. So we're dealing with the field called taxonomy. So taxonomy deals with, you know, identification. And identification can take place in different ways, and we're going to see how that can happen. For example, you know, just by looking at all the different kind of bacteria that are being grown in the laboratory. I don't know if you've looked at some of the petri dishes of your classmates or not, but you might want to just look at it because they are different. You know, we're all growing colonies, but they're all like different sizes, they're different colors. I think Hannah, yours was the one that was fluorescing, right? And Allie, you had those beautiful reddish pink ones, right? So there's a lot of variety there. Just by looking at them, that can help us to distinguish, you know, one from another, and that's the idea. That's the basis of identification. You know, to you know, use the features in order to be able to say that this one is different from that one. All right, the second part is called classification. This is where we can put related or similar you know, bacteria or prokaryotes into common categories, you know, common groupings. For example, you remember how we can use differences in cell wall composition to, put bacteria into two different groups?
Yes.
Okay, and what do we call those two groups? Gram-positive and gram-negative. That's right. So that's a really good example of classification, and that's a nice way that we can use to help us with our own identification that we're trying to do in the laboratory. And then the last item there is called nomenclature. So, that's about naming. Nomenclature is naming. So there's actually some rules about how, once you make you make a new discovery, you discover a new species, a new bacterium, how there's certain rules about naming it. Just for fun, how many new species do you think are roughly discovered each year? Like, it doesn't just have to be bacteria, it could be like any form of life. 1,000?
200.
200? All right, you guys got the computers. Why don't you use AI and ask AI about how many new species are discovered each year. What, you never use AI for? What's AI? Okay, who's got the number?
I got between 16,000 and 18,000.
Wow, 16 to 18,000 new species are identified every year. I mean, darn, that's a big number, right? That's bigger than any number you provided, right? And so, I mean, that's really amazing because, we think we live in, and we do live in a very advanced, high-tech world, right? You got all these satellites, we got these drones flying around, cameras are on, and yet there's tens of thousands of species out there that we haven't laid eyes on before.
Isn't there something about species at the bottom of Marianas Trench being maybe more undiscovered species than we have on the planet or something?
Yeah, I mean that's a good point there. A lot of these species are in areas of the world where they're not readily accessible, right? Where maybe humans haven't really adventured or reached yet. So, and some of these are rather big species too. They're not just the microorganisms. So knowing that, well, that's kind of exciting, right? Because that means like you could make a discovery, like over there, there's a species, and then you can then name it. All right, so we're gonna start, thinking about, let's delve more into taxonomy. Beginning with how to identify a species. So there's different ways that are listed here. Because we're dealing with microscopic organisms, we have microscopic examination. So in other words, just through observations, what do they look like? Remember, think back to our first lab session. You got the microscopes out. You used oil immersion to look at the slide of prepared bacteria. What did you see? Different shapes, right? Different shapes. There you go. And you did the same thing with your own bacteria a couple weeks ago, because that's a way of helping us to distinguish. So species have a defined shape that can be used for identification purposes. Okay, culture characteristics. I know this seems a little weird use of the word culture here, but in this context, we're talking about growth. Culturing means growing. Okay, how are they growing? Like in our case, in the petri dish. So we talked about some of those features already. Size of colonies. The margins. Color. Things like that. Those are culture characteristics. And then in a couple of weeks, we're going to do some biochemical tests. So this is related to metabolism. We're going to test our bacteria to see if they can ferment different kinds of sugars. So can they use glucose? Can they use lactose as a source of energy? And do they produce, as a result of fermentation, can they produce acids? Can they produce carbon dioxide? So those are all related to biochemical pathways, right? Nucleic acid analysis. So some of you are taking genetics, I think. Mallory, I think you're taking genetics, right? This is right up your alley. So you got DNA, and you also have, what's the other nucleic acid? You got RNA, right? And remember, DNA and RNA, they're made up of nucleotides. So, okay, who can recall the 4 kinds of nucleotides that make up DNA?
Adenine, guanine, cytosine, and thymine.
That's correct, yeah. And a lot of times people just remember the first letters, like A, T, G, and C. And then likewise, RNA is also made up of nucleotides. So what are those for?
AUGC?
A, U, and G and C. So you have uracil instead of thymine. So the analysis then refers to the order, what we call the sequence of those nucleotides that are found in either the genes or the products of those genes. Okay. All right. So in other words, the genes can help to distinguish one species from another. And then the last one is for organisms that can that are causal agents of disease. They induce symptoms that can also be used to identify them. Like think about influenza. That's a virus inducing flu symptoms. Fever, vomiting, sore throat. Just by those symptoms alone, you'll know whether you have a particular pathogen or not. Okay, so those are some different ways of that we can use to distinguish and identify microorganisms. All right, then we think about classification. So we're thinking about this time about groupings, based on those common features, and we're dealing with what's called phylogeny. So we're talking about evolutionary relationships here. So like, why is it that 2 or more species have the same features? Why do the bacteria have, similar colony color. Like, let's say you have a bunch of bacteria, they're all making yellow pigment. Like, why is that? Well, pigmentation is a product of enzymes. Enzymes are products of genes. So if they're making the same color, they have similar genes. So why is it then that they have the same genes? Well, because they inherited those genes from often common ancestors. Okay, so, you probably know, even within your own genetic familial relationships, similar hair color, similar facial features that you share with your siblings, your cousins, your nephews, your aunts, your uncles. Well, why is that? Similarity in genetics. Okay. All right. So that's the idea here, is that when we look at similar groupings, they often reflect genetic similarities due to similarities in their genetic relatedness. That's related to, that's evolution there. All right, just want to remind you about this. You don't have to memorize this one again. I mean, you've seen this so many times before. But, at the top, so we're looking at these hierarchical classification levels that are used here. And just as a reminder, when we talk about these scientific names, it's a binomial. So what do we mean by that? Binomial. Two-part names. Like we have Escherichia coli. Those are two words. Okay, so the first word is the genus. The second is the species, also known as specific epithet. Okay, so don't worry about memorizing this one. That's just for fun there. All right, here's something else that's of interest, and this goes back to how did scientists come up with the 3-domain system of classification? So, in other words, of all the living things on the planet, you can classify them into one of 3 domains, and those names they're on the slide there, although they might be a little small to read there. I don't know. Can you read that in the back? Okay. So on the left, what do you see, Zach?
Bacteria.
That's on the right. What do you see on the left?
Bacteria.
Well, this is a good eye test. Okay, so you got domain bacteria, domain eukarya, and domain archaea. Okay, and sometimes this is phylogeny here, so it shows the interrelationship of these different kinds of organisms based on evolution. Okay, so let's see if we can figure out what this is showing us here. Okay, so for example, over here on the upper right, you have animals, fungi, and plants. And so, in other words, where you see all these names, those are all living organisms that we see on Earth today. So all these are present on Earth today. Okay, what does it mean by the lines, these 3 lines, the lines for animals, fungi, and plants, all converging into a common point? What is inferred by that? That's right. They come from a common place. So in other words, remember, all these names are refer to organisms that are alive today. But as you follow the line away from the name, you try to imagine going back in time, in years. Like millions of years. So going back in time, millions of years, there was a time where scientists believe that there was an organism that was the common ancestor to modern day animals, fungi, and plants. So that wasn't yesterday or the week before, right? No, that was millions of years ago, where we shared an ancestor with a mushroom and with a fern plant. Okay, hard to imagine, right? Okay, so that's the concept here. And then, well, how did they? What's the basis for this idea? It sounds kind of weird. Well, it's based on looking at DNA sequences. DNA sequence for ribosomal RNA. So remember, what does a ribosome do? It makes proteins. Involves in protein synthesis. So all these organisms that you see listed here make proteins. They make proteins using ribosomes. They have ribosomal RNA as a component of their ribosomes. So remember, ribosomal RNA is made of RNA, of those 4 RNA nucleotides. Allie, what are those 4 again?
RNA? G, C, U, and A.
G, C, U's, and A's. And remember that there's more than just 4 of these letters that make up ribosomal RNA. Okay, so there's going to be like 1,000 of them. But if you compare the ribosomal RNA for plants versus the fungi, they're very similar. But then the idea is that as you move away from any particular organism, they get more different. For example, the ribosomal RNAs for these 2 will be more similar to each other than the ribosomal RNA for the diplomonads versus the aqua fecunda. Does that make sense? All right, and then what's shown in here is that you get the 3 main groupings based on their ribosomal RNA sequences. Okay, so looking at this figure here, remember you are an animal. Are you more similar to a bacterium? Or are you more similar to archaea? It is archaea. That's right. You are more similar to archaea here than you are to bacteria. All right, now here's something interesting. See how all the lines converge to this single point? Right here? What does that mean?
Because the common ancestor for all of this.
That's right. So the thought here is that there was an original progenitor for all of life. Okay.
But to what extent is this true, and what is the scientific evidence of backing that up?
Yeah, it's all. I mean, we're going to see another slide here, but we have our ribosomal RNA data, And then the next slide is going to show us a similar kind of phylogeny based on ribosomal protein amino acid sequences. So you can actually look at like two different things, and it kind of gives you the same result here. Here's something interesting about, this original source of life on Earth. Here's something wild. Let me draw this one out because it's pretty crazy here. Okay, so try and imagine this is Earth. And remember that there are planets in the solar system here. So this one over here, you pretend this one is Mars. And you may have seen those pictures of the planets with all those large asteroid impact craters on those planets. Almost like bombs were dropped on them. So there must have been a time when there was just a lot of asteroids hitting the planets. And one thought is that one of these asteroids impacted Mars to such a degree that it caused like a mini explosion that launched pieces of Mars out of its orbit, basically, into the solar system. And believe it or not, scientists actually discovered a piece of Mars on Earth. Can you imagine that? The impact causing a piece of another planet to land on our own planet. Okay, how's that related to this picture over here? One thought. Needs further testing. One thought is that this original source of life on the planet, on our planet, may have come from outside of our planet. Or maybe from Mars. So in other words, maybe there was life on Mars at one point, and then that was introduced to Earth, where it flourished and developed into everything that we see today. If that's true, what does that mean about you? It means you're an alien. Kinda. So needs further testing, right? But that's a big question about how did life begin, right? Okay, so here's the other piece of evidence for that 3-domain system. This one involving the order or sequence of the amino acids that are found in the proteins of ribosomes. So this time, instead of just looking at those 4 nucleic acids, 4 different kinds of nucleotides, this time they're looking at, anyone know roughly how many amino acids there are? The main amino acids?
Is it 14? It's I'm not sure.
About 20-ish, yeah, 20 main amino acids there. So you have a little bit more to work with now. And they came up with this kind of a phylogeny. I know it's really hard to see there in the back, but eukarya is over here on the right, so that's where we would be. And archaea is right there, right next to us. And then at the top, that's bacteria. So it kind of reinforces the idea that, we are more similar to archaea than we are to bacteria. Okay. All right, so that's part of classification, and even though we are, the eukaryotes are very similar to archaea, the ribosomes of archaea are actually more similar to those of bacteria. So don't worry about everything else that you see on the table here, because we've actually seen most of this stuff before. So focus on differences in terms of ribosomes. All right, what do the numbers and letters mean? Well, the letter refers to the unit. It's related to a sedimentation coefficient. So you're familiar with a centrifuge? You have the spinning device similar to the spin cycle on a washing machine, but you can for the instrument in the laboratory. The larger the sedimentation coefficient means that the ribosome is physically larger. It's got more mass than the smaller numbers. So the ribosomes of bacteria and archaea are structurally smaller. But there's also other differences there too. Differences in terms of the ribosome RNAs and also the ribosomal proteins that make them up. And fortunately for us, this difference in the ribosome means that there's actually some chemicals that can prevent bacterial ribosomes from functioning that do not harm our ribosomes. So what do we call those chemicals that prevent bacteria from making proteins and growing? Close, we call them an antibiotic. Okay, so this is actually one target for antibiotics. It's the ribosome because of this difference that we see. Okay, so we talk about identification, classification. We're moving into naming, nomenclature. At the top of the slide, we have a book. That was named after Berge, so it's called Berge's Manual. And this is, think of it as like a reservoir for all knowledge known about prokaryotes, bacteria and archaea. So everything that's known about them, how do they grow, what do they look like, what color are they, what's their oxygen requirement. All that information is compiled into a single source we call Berge's Manual. I believe it's online. Okay, so at the end of the semester, after we're done doing all of our biochemical and other kind of stains and testing of our bacteria in the laboratory, each one of you will take a look at what's known about your bacterium from this source here we call Berge's Manual. So I'll help you out with that once we get to there. So just kind of keep that in the back of your mind as at the end of the semester there. And then earlier we learned that there are rules for naming, and those rules can be found in the International Code of Nomenclature for Prokaryotes. Well, I like pictures. I hope you do too. So we're going to take a look at some examples here of different photos showing different ways that these prokaryotes can be identified. So we're going to begin with differences in how these organisms appear when viewed under the microscope. Well, we've already seen pictures of gram stains, purple and pink bacteria. So now we see something a little bit different here. For example, on the right, we have an example of bacterium streptococcus that's been stained with what's called the acid-fast stain. So we're gonna actually do this one in a couple of weeks here. So this stain is actually helpful in identifying a bacterium called Mycobacterium, the causal agent of tuberculosis. And hopefully none of you have that growing in your petri dish, but we don't know until we do the stain. But the concept here is that this bacterium has a thick outer covering made up of lipids that makes it difficult to stain. So we actually have to use a little bit of acid to penetrate that exterior in order to get the stain inside of it. So you're going to see, some like in this case, green and red coloration there for that stain. And then just looking at under the microscope, you can also observe larger items like we see on the left with our roundworm egg. So none of you are growing roundworms in the petri dish, so you don't have to worry about that one. And then over here on the right, we have a bacterium called Neisseria, and that's the causal agent of a disease we call gonorrhea. So we'll see what's happening there too. Okay, so we've already done this initial step with your bacteria, looking under the microscope. We've done the gram stains. We'll probably do one more test there, a couple more stains, and so on. All right, so other than the microscopic observation, we can also make more detailed notes about how our bacterium is growing in the petri dish. So here's some fun characteristics here. For example, we have certain bacteria that are associated with color, like red for Serratia marcescens. So what's kind of interesting about Serratia is that the color is temperature dependent. So if it gets too warm, it won't make the color. So that's an interesting one. So it's what we call a temperature sensitive gene there. And then I already mentioned about Pseudomonas aeruginosa, because that's the one that often infects burn patients. Remember that one picture of that operating room where you had contamination throughout the room? That was from Pseudomonas aeruginosa. There are a bunch of other species of Pseudomonas that make pigmentation too. So Hannah, your fluorescent one might be a Pseudomonas too. I don't know. Whenever you get a chance, you should get the opportunity to check out Hannah's bacteria, because I think you've all seen fluorescence before, but have you ever seen like a living organism fluorescent? All right, and then we can use growth on different kinds of media. So that's like the stuff inside of the petri dish to help us understand what the bacteria are able to do. So one thing that I want to do, I'm trying to get the school to order this stuff for us, but there's a media called blood auger. So you know what auger is? That's the solidifying agent. And now it has like real blood in it added to it. Don't worry, it's not human blood. We're not looking for donors here, but it's actually from sheep. Sheep blood. So the idea here is that there's certain bacteria, like the streptococcus here, that when it causes disease, it can actually break down heme that's in blood. So, blood is red. So when it breaks it down due to beta hemolytic activity, it'll turn that red into clear. So if you see that in the petri dish on blood auger, that gives us an idea that we have streptococcus pyogenes. And then last thing here is that we will be looking at lactose fermentation. Because people, and these are designed for E. coli detection. There's special auger that where the E. coli will form pink colorations on. So this is not, McConkey is not the auger that you are using. So if you see pink, that does not mean that it's E. coli. So it's only on this McConkey auger that the pink is an indication of E. coli. All right, I think this will be a nice place to stop. Any questions? All right, then. Everyone have a great day, and I will see you on Friday.